Hello everybody. Today, as part of our online video education, we are going to discuss, learn about sexual reproduction in flowering plants for class 12th. At the end of the session, you are supposed to be able to define Microsporogenesis, syncarpus gynoecium, apocarpus gynoecium, as well as understand the structure of TS of flower, VS of Microsporangium, as well as Meghas Parangium. Also, every student is able to reflect on the process of Microsporogenesis. With this learning objectives in mind, let us start chapter number 2, Sexual Reproduction in Flowering Plants. The sexual reproduction, as we discussed in the previous chapter, is a reproduction that takes place with the help of gametes, male gamete and female gamete. In a flowering plant, the male gamete is the anther, and male gonad is the anther that produces pollen grains. So pollen grains are the gametes, and the female gametes are the ova or ovum singular, which is produced by ovary. So the reproductive structures of flower are called androecium, male structure, and gynoecium, female structure. So you can see both androecium consisting of stamen and gynoecium consisting of carpel. The male reproductive structure, androecium, consists of stamens, overlap stamens, a round of stamens, and each stamen has got a anther, a small bag-like structure, and a filament. So you can see this in the vertical section of the flower. And a stamen has a slender stack called filament. At the tip, it contains a bilobed structure. So the anther is bilobed structure, as you are able to see in this picture. Generally, anthers are bilobed, that contains two theca, dithecus. Di means two, theca bag-like, separated by longitudinal groove, as you are seeing. When we take transverse section of this anther, we can see it consists of four-sided microsporangia, two in each lobe. So you are seeing the same four theca here. The microsporangia develops into pollen sac. Inside this pollen, grains are present. So you can see the structure here, pollen and the filament at the center. So anther having bilobed, and the pollen sac containing pollen mother cells. Pollen mother cells will be always diploid. So the PMCs, pollen mother cells, undergo meiosis and form tetrads of pollens. And you can see in this picture a stamen having anther and a long stack-like filament. And in the anther, four-lobed structures. And inside the pollen sacs, formation of pollen grains will take place, and on maturity of the pollen, they get the dehiscence or dispersal of pollen grains will take place. Now let's go to the structure of Microsporangia. Microsporangium, as you are able to see in this picture, is surrounded by four layers. The external one, outer one is epidermis. It's a single layer, and its duty or function is to protect the microsporangium. And second one is endothecium. It's again a single layer of cells, characteristics of dehiscence. It helps to release pollen. The dehiscence character is due to hygroscopic nature. And there are 3 to 4 layered middle layers, again which are protective in nature. And the final one is tapetum, innermost layer of Microsporangia. It plays a crucial role as it provides nutrition to developing microspores. You can see the structures here. So the pollen grains, we can see the epidermis, we can see the tapetum, the inner layer, and the tapetum provides nutrition to the growing pollens. And how does this happen? Through a process called Microsporogenesis. It is the process of formation of microspores or pollen grains from pollen mother cell by meiotic division. So remember this Microsporogenesis or Microsporangiogenesis. The microspores formed are a cluster of 4 cells called microspore tetrad. As the anther matures, it dehydrates and the microspores will get dissociated, develop into pollen grains. Now let's see the structure of the pollen grain. A pollen grain normally spherical in shape, 2 layers it has got. One is outer exine and the inner intine. The exine is made up of an organic compound called sporopollenin. Please keep remembering this word, sporopollenin. Because of this presence of sporopollenin, the pollen grain will be very tough. And then it is very hard, resistant organic compound, and no enzyme is known to degrade this as on today. This exine has a prominent pore called germ pore. As the pollen grain matures, it contains 2 cells. One is vegetative cell and the other one is generative cell. The generative cell divides mitotically into 2 male gametes. As you are saying, haploid nucleus, young pollen grain undergoing mitosis, forming mature pollen grain. You can see the outer layer, outer exine, inner intine, and then 2 nuclei, one generative nucleus and a tube nucleus. And now let's see the structure of gynoecium. A gynoecium is the female reproductive part of a flower. It consists of a pistil, and gynoecium, if it has got a single pistil, then we say it is monocarpellary. And if it consists of more than one pistil, then we say multicarpellary. And in some flowers, the gynoecium will fuse with pistil, and then we say it is syncarpous. Syn means mixing. And in apocarpus, of course, the gynoecium will be having free pistil. And you can see the monocarpellary. Mono means one carpel. Example, pea, beans. And then multicarpellary, where it is syncarpous, it is fused. Tomato and cucumber. In apocarpus, of course, we have got examples wherein lotus, vinca, these are the examples. And let's see the structure of pistil. Pistil has 3 parts, as you are able to see in this picture: stigma, style, and ovary. So you can see on the extreme one, stigma, style, and ovary. Stigma will have bifurcations, and style is a slender tube. And then of course ovule will be like a broader one, pot-shaped structure. Stigma is the tip of the pistil that serves as a landing platform for pollen grains. And style is the elongated slender part beneath the stigma that helps in making a path to the pollen grain to reach pollen, to make a pollen grain to reach ovule. And ovary is the basal bulged part of the pistil. The cavity present inside the ovary is called locule. The cavity which is present inside the ovary is called a locule, ovarian cavity. The Megasporangia, commonly called ovules, arises from placenta. Placenta is again a tube. The number of ovules in ovary may be 1 like in wheat, paddy, mango, or many like in papaya, watermelon, orchids. Now let's see the Meghas Parangium or the ovule, the structure of ovule. The ovule is a small structure attached to the placenta with a stalk called funicle. Look at that funicle here, elongated one at the bottom, young ovule, and you can see 2 sides, micropyle end, and the other side embryo sac is present. And you can see the body of the ovule fuses with the funicle in the region called hilum. Look at the part hilum, right? And each ovule has 1 or 2 protective envelopes called integuments. So integuments are protective envelopes. Integuments encircle the ovule except at the tip. The small opening present at the tip is called micropyle. See in the diagram, micropyle, which is the small opening at the bottom. Opposite to the micropyle end is chelagel end, top one. Look at the top, chelagel end. It represents the basal part of the ovule. So you got two ends in this, micropylar end and chelagel end. And within the structure, you got antipodals, egg cell, secondary nucleus and synergids. Okay. And the anatomy, when you see the anatomy of the angiosperm ovule, the integument encloses a mass of cells called nucellus. Nucellus is very important as it serves as a nutritive layer. These cells have abundant reserve materials. The embryo sac of the female gametophyte is located in the nucellus. A ovule generally has a single embryo sac formed from megaspore through reduction division. So in this vertical section, you can see chelagel end, micropyle end, nucellus, embryo sac, ovum, synergids, funicle, outer integument, inner integument, antipodals. Now if you count the numbers, you got on top 3 antipodals. Centrally there are 2 nuclei which are central part, and then down at micropylar region you got 2 synergids and a ovum. So totally you got 8 nuclei, 3 plus 2 plus 3. Now with that we will go to the next session. Thank you.